Next, we will have Emily Oldani.
Hi, so I'm one of the researchers in Dr. Sunil Kumar's lab over at the University of Denver, and we don't do anything clinical, but we are in general looking to develop lead compounds to be good drugs to treat Parkinson's, Lewy body dementia. Currently, we started an Alzheimer's project, and for this, we're going to focus on our Huntington disease research. So protein is a biomolecule that can aggregate or clump in many neurologic diseases. So it starts by being monomeric, which is just one copy. That becomes oligomers, which are several copies. That can become protofibrils, which then become those inclearable, insoluble amyloid fibrils that lead to diseased brain cells that lead to that phenotype that you see in neurologic diseases. Our drugs aim to stop this. So if we're just looking at it from aging and neurologic diseases, there's a healthy brain and a diseased brain. You have that atrophy of the brain overall, neurons start to fall apart and die. These include many different ones, but our research is specifically focusing on Parkinson's disease and Huntington's disease for the scope of this grant. One thing that I do really want to let you guys know is essentially any biomolecule is a Lego. And the way Legos interact by having specific structures that can allow them to perform specific functions and interact with certain other shapes, that's kind of a unifying principle behind a lot of biochemistry. So specifically, our drugs are designed to fit really well with the shape of proteins that are involved in different neurologic diseases. So there's these things called beta sheets that can form those really insoluble things. There's also a lot of things called alpha helix, and our drugs are the right shapes to go with that. You have to have folding for each protein. If it doesn't fold right, it's not going to be able to perform its job. So that's what a lot of people have talked about so far today with Huntington protein not really folding into a uniform shape or the correct shape. Certain shapes can also become something that can act as a sponge and siphon out anything that would be really helpful in a cell, like RNA that's trying to go produce protein that's functional in a different region of the cell, and proteins that are trying to do their own job and sequester them from being able to perform their normal functions. And then a lot of them can become too strong to break up. So our drugs aim to target that, and they're kind of the best of both worlds because you often have small molecules that have very low specificity, but can get into almost any region of the body or cells or organs, or antibodies, which are very, very specific, but they're very, very big. So we're trying to balance between too small and specific. Our drugs fall a lot closer to the small molecule side, and then they're a lot cheaper and more specific and easier to control than sRNA, which is the current main direction that a lot of Huntington disease research is going towards. So once again, just really bringing home that point of Huntington protein aggregates by being monomeric and then oligomeric insoluble fibrils. We want to disrupt that with the addition of our drugs by breaking it back into just oligomeric and monomeric protein. So we're targeting those specific structures, and then we kind of group this goal into two sub-aims, which would be preventing the clumping, preventative care, or inhibiting, and then disease treatment, which would be breaking up those clumps, which would be just a treatment strategy. So the aim of our drugs is to return the brain from being something that is becoming atrophic to something that's a lot more healthy by breaking up those protein fibrils. And then we have a general therapeutic pipeline that our lab works through. So we have HEX cells, which are just a particular type of human cells that you can grow really well in a dish in lab environments, and you can tell them to do all sorts of very specific things. We use them to express proteins that are involved in Huntington or Parkinson's disease with this fluorescent tag on them so that we can find it always being bright green and know exactly where it is and if it's aggregating or not. C. elegans, which is a flatworm that is really sensitive to neurologic diseases that humans suffer from, but it's really nice because they're small, they're see-through, and they only live for about 2 weeks. So you can get a lot of information out of them. Primary neuron culture, and then mouse models. With our therapeutics for Parkinson's disease, so far we have hit the stage of working in mice, and with Huntington, we just reached the point of working in C. elegans. So one thing to remember is our Parkinson's disease research began 8 years ago, and our Huntington's disease research began last year with the start of this grant. Just going through, you start to narrow that target so you start with a large library of molecules, and then you narrow it down as you find more and more leads. So our Parkinson's disease and Lewy body dementia are in that phase, and then our Huntington's disease model is a lot slower behind or further behind, but it is moving forward. We do believe that we'll be able to move towards using our molecule in human trials for Parkinson's in the next year. But we've screened over 600 drugs now with our Huntington's disease model and are working on validating our hits and beginning our worm work. We are working on moving forward with continued funding in order to continue to advance our Huntington project. So within the next couple of years, we aim to be screening these drugs in the worms, getting all of the information we can out of them. So far, we've just been able to see, yes, it looks like it's working visually by seeing if there's aggregation or not. Not any sort of health monitors that you can do in those worms. And then we're working on further refining the drug pharmaceutical properties for the best efficacy and the most optimized safety, just by seeing what sort of products would come from our molecule being taken as like an oral drug or intravenously. Over time, we do want to move forward with a Huntington's disease mouse model, as well as kind of focus on making this project become more of a mature project that is supported by a full lab instead of just me in our lab. And then we'd like to move forward with some machine learning, since we have a large data set, and would like to further refine the information that we're getting out of it instead of just using it to find out what an actual hit is. So I'm going to give you guys some of our preliminary data so far. So this is prevention. So this is if we treat our Huntington's disease expressing cells with our molecule and then induce them to aggregate. And then it's just measuring how aggregated they get. So white is going to be got worse than baseline. Yellow is going to be not great. And then pink is going to be, it significantly reduced the aggregation. So one box is one drug. That's how many I've screened so far. That's how many our chemists have made. It's 602 currently. And then when we flip that for the treatment score, which would be treating or inducing the aggregation and then treating with that molecule, we also see many hits. When we sum those together, we can refine that to get the best molecule to both prevent and treat. So keeping the Huntington protein from aggregating, but also breaking it up and keeping that from seeding further aggregation as well. You can see there's fewer hits, which makes it more feasible to move forward with. And then this is just what a healthy cell on the left looks like. So you can see there's not really bright spots of green in that. The blue is the nucleus. We expect to see that. It's just marking where it is. And then a healthy worm where we just see green all the way through. In our disease models, so we have a lot of those bright spots, which are called puncta. Those are those aggregates. And we see that in the worms as well. And then those fibrils are just from protein in a test tube. We can get it to aggregate. When we treat it with our drug, we get this return to looking a lot more like the healthy model in the cell line, the worm line. And then we don't really see aggregates. We just see broken up clumps of oligomer and monomeric protein in the treated. And then this next slide is kind of the most exciting. We just did some overnight imaging to see if we can watch those large droplets dissolve in real time. So it looks like this, but if you focus in on the middle, the droplet in the lower quadrant is going to dissolve and become diffuse, and you can see it stop being an aggregate and something that's spread throughout the cell, not necessarily acting as an unclearable thing and not likely causing that cell to die. And with that, I do want to thank the lab that I work in, because science isn't a just one person job. We have a lot of people that work on this project, even though I'm the only one doing the testing. So we got the use of an overnight microscope from a collaborator and her graduate student. We have 6 chemists that are working on making all of those drugs for me, my PI, Dr. Sunil Kumar, and then my undergraduate, Naya, has been working with the worms for me. With that, can I take any questions?